Hi, welcome back. Michelle Glass here. We are still involved in our conversation of the kidney. So we are still in our chapter 26, lecture 26 series. And our topic here is the nephron loop, which can also be referred to as the loop of Henle. Now the nephron loop has the important job of reabsorption. So remember, this term reabsorption is talking about returning material back to the body. So that means it leaves the kidney and makes its way ultimately into the bloodstream to be back in circulation. The nephron loop has both a descending and an ascending limb or loop to it. And we will see different things happening in each of these. In the descending limb, we're going to see the reabsorption of water. And in the ascending limb, we're going to see the reabsorption of sodium chloride. Okay, so remember that we had at the end of the PCT only about 30 to 40% of the initial filtrate left as our tubular fluid. That fluid is going to make its way moving down the descending loop of Henle, and we will see that the membrane of the descending loop is permeable to water. So we will see water being reabsorbed here. So we're getting about 25% of the water from the original filtrate being absorbed here, so that ultimately we're getting about 85% total of the water that was initially filtered out being reabsorbed. As we get to the base of this limb, all of those solutes are going to become super concentrated because so much water has left. When we are moving up the descending loop, we will see that this loop or limb is going to be permeable to sodium chloride and not water. So because you have super concentrated your sodium chloride at the base of the loop of Henle, as it moves up the ascending limb, we will see that sodium chloride is going to move with this concentration gradient from high to low. Now notice that you have opposite movement as we're looking at this limb. So because we have movement close together, down and up, we have what's called the countercurrent multiplication happening here. So this opposite movement that's happening creates a really strong concentration gradient. So that what we see happening is in that ascending loop, we've pulled out all the sodium chloride, which then is going to help all that water get pulled out from the descending loop. Right? So you're creating a hypertonic peritubular fluid, which is going to pull water into that peritubular fluid. So we're enhancing the pull of water from the descending loop because we have super concentrated the peritubular fluid with the movement of sodium chloride out of the ascending loop. This is establishing what's referred to as the medullary osmotic gradient. So in the medulla, so that is those renal pyramids, we're going to see the peritubular fluid. It's going to be full of sodium chloride. And this is going to help pull the water out of that descending limb of the loop of Henle, as well as pulling water from other areas such as the distal convoluted tubule and the collecting duct as needed. And that's it for now.